Hello friends, on behalf of Team Nursing Extra Mileage, I welcome you all. Today I am going to discuss a very important topic, that is human body organization at the cell level. For that, we need to understand first what is a cell. So it is the smallest functional unit of body. There are two types of cell, prokaryotic cell and eukaryotic cell. Eukaryotic cell we will discuss in this chapter mainly the eukaryotic cell. So eukaryotic cell, the structure is made up of a cell membrane which is a covering outside the cell, a nucleus inside the cell, and surrounding the nucleus there is cytoplasm and organelles. Let me draw a cell and make you understand easily. So suppose this is a space cell. So the outer covering, this is called the cell membrane, the covering, and inside the cell there is a nucleus, and surrounding the nucleus there are cytoplasm remain. This is cytoplasm surrounding the nucleus where many organelles remain. So this is overall the structure of a cell. Now we will discuss each part separately. So first thing which is coming is cell membrane. Cell membrane is consist of protein, lipids and carbohydrate, all three elements. Here it is consist of proteins, lipids and carbohydrates. So which are the lipids? This is the glycolipids and cholesterol. These are the lipid part. Cell membrane is only 7.5 nanometer thick. So lipid part are the phospholipids and cholesterol. Phospholipids are the headed pin size. Outer part is hydrophilic and inner part is hydrophobic. So what is the meaning of hydrophilic and hydrophobic? Hydrophilic is water soluble and hydrophobic is not soluble. So what is the meaning of outer part and inner part? Outer part means if it is a cell, this is the outer part. So if a phospholipid is remaining, the outer part is water soluble and the inner part is not soluble. Cholesterol molecules remain in between the phospholipids. So two phospholipid molecule and in between remains cholesterol. Lipid layer is semi-permeable and allows only fat soluble substance, that is oxygen, carbon dioxide and alcohol to pass through it. Water soluble substance cannot pass. Coming to the protein part of the cell membrane, it is situated on either side of the central lipid layer. There are two types of protein. It is important, integral protein and peripheral protein. So integral proteins are also known as transmembrane proteins. Why it is called transmembrane? If this is a cell and this is a cell membrane, so transmembrane proteins means these proteins remains throughout the cell membrane, transmembrane, and they are tightly bound with the cell membrane. They pass through entire thickness of the cell membrane, which I have already drawn in the figure. Peripheral proteins don't penetrate cell membrane but are loosely bound with the cell membrane. So integral proteins binds hormone at its outer part, which brings conformational changes inside it and it becomes active. So if it is a cell, integral protein is situated in the cell membrane. Here at the outer part a hormone binds and it brings a conformational change inside the cell membrane, inside the cell also, and the cell becomes active. The integral proteins helps in transport of various substances. Integral proteins can also act as cell adhesion molecules which helps to attach cell with extracellular matrix. Coming to the third element, that is carbohydrate. It remains as thin loose covering over membrane. When remain attached with the protein, it is called glycoprotein, and when remain attached with the lipid, it is called glycolipid. So the carbohydrate part, suppose it is the carbohydrate part of a cell membrane, if it is remain attached with a fat part, it is called glycolipid, and with a protein part, it is called glycoprotein. So glyco part is the carbohydrate part. They are negatively charged and don't permit negatively charged substance to move in and out. As they itself are negatively charged, So negatively charged particle minus particles cannot move in and out. They are not allowed by the carbohydrate part. So role of cell membrane in transport. If it is outer covering, that is called cell membrane of a cell, how it helps for transport of substance from the cell and inside the cell. So if a substance is coming near the cell membrane getting invaginated means inner side invaginated, and substance getting attached to it, they are forming a vesicle, and vesicle gets detached from the cell membrane itself and they are released inside the cell. This process is called endocytosis. So substance coming outside from the cell and inside the cell. Exocytosis is expelling out some substances from inside the cell to outside. So the procedure is same. It is getting bound with some vesicles, the vesicle getting attached to the cell membrane, and then it is throwing out substances from the cell. This process is called exocytosis. Now the description: molecules enter cell by endocytosis, which I have told earlier. When it is fluid, it is called pinocytosis, and when it is bacteria, it is called phagocytosis. So phagocytosis and pinocytosis are the two process of endocytosis, means bringing the substance inside the cell. Exocytosis is method to expel out substance from the cell, which I have told earlier. Now coming to the nucleus. At the first slide I had drawn a picture that if it is a cell, the outer covering is called cell membrane, and inside it remains the center, it is called nucleus. So it is present in the cell which divide and produce enzymes. So prokaryotic cells does not have nucleus and eukaryotic cells do have. Here we are discussing the eukaryotic parts only. Prokaryotic cells are the older cells that doesn't have any nucleus. Most cells have one nucleus, but few cells like skeletal muscle cells have multiple nuclei. Nucleus may be single or multiple, but there are the exceptions where multiple nuclei are present. Nucleus have same like the cell having a cell membrane, nucleus is also having a nuclear membrane. This is the nuclear membrane, and it is double layer and encloses the fluid called nucleoplasm, and inside the nucleus another thing is present which is called nucleolus. In the center it is called nucleolus, and surrounding the nucleolus there is nucleoplasm. So three part: nuclear membrane, nucleoplasm and nucleolus. These are the three parts of a nucleus. Nucleoplasm is gel like and contains DNA which is made up by chromatin. Easily to understood. One or more nuclei present in each nucleus. Like a cell may contain more than one nucleus, nucleus also may contain more than one nucleolus. Nucleus controls all activities of the cells. For cell to divide, the cell has to depend on the nucleus. So this is a cut section of a nucleus. So there is nuclear envelope, that is nuclear membrane. Here at the center it is nucleolus. This is nucleoplasm, that is the surrounding matrix, and there is chromatin. Now coming to the cytoplasm and cell organelles. So it is a cell, whole the thing drawn, it is a cell. The whole thing is cell. It is a whole cell. So this is outer covering is the cell membrane. As we already seen, this is like written plasma membrane, and inside it is the nucleus, which I can see. This is nucleolus. Inside the nucleus there are cytoplasm surrounding the nucleus, which I have told earlier, and in the cytoplasm there are many organelles. They are mitochondria, the Golgi apparatus, the rough endoplasmic reticulum. There are also smooth endoplasmic reticulum. Here it is smooth endoplasmic reticulum. They are the cell organelles surrounding the nucleus and situated in the cytoplasm. This is the cytoplasm. Here the organelles are situated. Coming to cytoplasm, cytoplasm is the liquid present in the cell. There are various structures in the cytoplasm of a cell known as organelles, which I have told earlier. That is endoplasmic reticulum, Golgi body, ribosome, mitochondria and different filaments. Coming to one by one. The endoplasmic reticulum, it's made up of tubules. It is of two types, smooth endoplasmic reticulum and rough endoplasmic reticulum. So what is smooth? What is rough? When granular ribosome is attached with endoplasmic reticulum, it is called rough endoplasmic reticulum. It is having a rough appearance. Ribosome does not get attached with smooth endoplasmic reticulum, so it is appearance is smooth. Rough endoplasmic reticulum deals with the protein synthesis, very important line, and smooth endoplasmic reticulum deals with the synthesis of lipid, steroids and cholesterol. These two lines are very much important, the function of endoplasmic reticulum. Now coming to the Golgi body, except RBC, it is present in all cells. It is of two phase, cis and trans. It is concerned with processing and delivery of substance like proteins and lipids to different parts of the cell. So it is having a transport function. Membrane bound vesicles. Some vesicles also present in the cytoplasm like phagosome, solid foreign material, that is bacteria ingested by cell. In earlier slide we have seen pinocytic vesicle, that is fluid filled vesicles, and exocytic vesicles to throw out substance from inside the cell. There are lysosomes also, which is membrane bound spheroidal bodies containing hydrolase enzyme. So these vesicles are nothing but some particles which remaining inside the vesicle and by which their name is being determined. If it is fluid, it is pinocytic vesicle. If it is bacteria, it is phagosome. If it is enzyme, it is lysosome, like that. Coming to the mitochondria, it is called powerhouse of the cell. Why powerhouse? Because by metabolism, ATP, adenosine triphosphate, ATP being synthesized in the mitochondria, ATP, adenosine triphosphate. In next slide, we will also see what is ATP. ATP. It is having outer membrane and a matrix called calcium, RNA and DNA. So mitochondria. This is a cut section of a mitochondria. Mitochondria also having an outer membrane and an inner membrane, and inside mitochondria there is DNA, ATP synthase enzyme which produces ATP, like that, and matrix. This is a cut section of a mitochondria. Mitochondria is the chief site of TCA cycle, tricarboxylic acid cycle, a part by which metabolism occurs, electron transport chain, and it releases energy from ATP. Now the cell extensions, they are projections from the cell. If it is a cell, they are the projections from the cell. So they are cilia, flagella and microvilli. Cilia moves together in a particular direction, they move together in a particular direction and thus helps to remove secretions of the trachea. Trachea means the air pipe. The secretions are removed by cilia. Now coming to the transport across the cell membrane, how the transport occurs. So there are two processes by which the transport occurs. One is called simple diffusion and another is called facilitated diffusion. From the name we can see that simple means simple. So they are without any helper protein, no helper. It occurs by its own. So it is called simple diffusion. And facilitated diffusion means some helper protein is helping to get the diffusion done. So if it is the inside the cell, if it is outside the cell, the particles which is coming and directly going inside the cell without the help of a helper protein is called simple diffusion. And in the facilitated diffusion, this is inside the cell, this is outside the cell, this is inside. So if any particle is coming, they are taking a helper protein, channel protein and going inside the cell. So this is nothing but a helping hand and this is having no helping hand, simple diffusion. So simple diffusion is applicable for lipid soluble substances like oxygen, carbon dioxide, etc. Facilitated diffusion need carrier protein. As it needs a carrier protein, so the process is faster than simple diffusion. Another type of transport called active transport, where movement of substance occurs against the chemical or electrochemical gradient. So the terminology is active, means against the electrochemical or chemical gradient. Sodium potassium ATPase pump is an example of this. So this is a third variety of transport across the cell membrane. Hope friends you are able to understand. Thank you. We will meet in the next lecture.